All right, welcome back to the lab. I got some raw honey here. We're going to test this, do some standard plate count media analysis, and see how many bacteria per, we'll say milliliter, are in raw honey. This honey is 1 year old. Honey does not go bad. This stuff is slightly crystallized. But bees collect water from different sources. I've seen them on manure piles. You would think that raw honey would have a lot of bacteria in it, and it's just inhibited by the high sugar content of the honey. So like if you, for example, harvested honey and it wasn't quite dehydrated enough, people's honey goes bad through a fermentation process. So bacteria that's transmitted from the feet and mouths of the bee are here. It's just a matter of the bees dehydrating that to the point where the bacterial cells cannot metabolize and proliferate or multiply. So I expect to find some bacteria in the honey. We'll see how much is there. And so the raw honey, some of the bacteria and the fungal spores, pollen and stuff like that, that's what people tout as helping their allergies. So if you buy raw honey from a local source, that pollen especially I would attribute to that's present in the honey would potentially help your allergies because you're exposed to that allergen in the honey at a low dose, which could then, potentially kind of over time reduce your allergic response to those local pollens in your area. Now the results from the honey experiment with the dilution, I didn't really show much growth at all, and with honey there really shouldn't be a lot of bacteria, but there could be endospore forming bacteria in there. So what I'm going to do is I'm just going to take a little bit of the honey and use a loop here and put it into a broth tube, and hopefully then I can at least grow something from the honey and then put it on a plate and take a look to see what types of bacteria are growing there. But basically the raw honey shows that there's not a lot of bacteria in the honey. There was some, but this isn't going to give us counts, but we'll be able to see what types of bacteria, maybe just how many different types of species are in the honey. So I'm gonna get a loop red hot here, sterilize it, and I'm just going to inoculate this test tube, and then tomorrow, if there's turbidity, that'll indicate growth, and then I'll be able to plate that and see if I can isolate some bacteria. So I got a little drip of honey here. And we'll let that sit overnight. So there's, mix that up a bit. Looks good. Alright, so this is the honey sample that I took, and I put in just a broth, nutrient broth, and now what I'll do is I will actually put this on a plate doing a streak plate technique, and we'll see if I can isolate bacteria, and then we can pick out some bacteria and do some staining. Most likely, from what I've done already, they're going to be gram-positive rod-shaped bacteria, most likely from the genus Bacillus, which are endospore-forming bacteria. So the endospores can basically just hang out in the raw honey, not cause any harm to you. But they hang out, and then once they're in optimal conditions, they germinate and then grow. So there is turbidity indicating that there is bacterial growth. But when I did the dilutions of the honey like I did for the milk, I really didn't have anything countable. So in general, raw honey usually doesn't have much bacteria in it. But in some cases I've read there can be lots of bacteria, but this is not going to give us a count, but we'll be able to plate this on nutrient media and see the different diversity of organisms in the honey, hopefully. So again, this is just going to be a diversity test, not going to get a bacterial count from the honey. We're just going to see what's in the honey. And just for fun, since I have some extra media, I've got blood auger. This is the same as our general purpose tryptic soy auger that we use often. This will grow pretty much anything. Blood auger, there's sheep's blood in it. Just for fun, we'll put it on there. ESN methylene blue again, looking for the gram negatives. And then I have a mannitol salt auger plate, so this is looking for salt tolerant organisms. So in honey, I doubt there'd be anything growing that's salt tolerant, but just for fun, see what grows, if anything. But I'll put the honey on 3 different TSA plates just to have some replication, and see what it looks like tomorrow. There's turbidity in this tube that I put a loop full of honey in yesterday. So there's bacteria growing in here. And we will see what it looks like. So the goal when you're doing this type of plating method is to spread the bacteria out so that you separate single cells so they grow as isolated colonies. So like those colonies that you could see on the milk plates, they'll grow as individual, and you can then pick those off and you can identify them if you wanted to. So I flame the loop to sterilize it in between each time I do the streaking in order to kill the bacteria that's on there, so it does reduce the number of bacteria as I spread them around the plate. So I should definitely have growth on TSA because this broth is tryptic soy broth. It's the same media, except this media has agar in it. Agar is a solidifying agent, kind of like gelatin. We use agar because agar isn't digestible by most bacteria. So some organisms can actually use gelatin as a carbon source for growth. This blood auger looks for what we call different types of hemolysis, which is the lysis of red blood cells. Again, it should, if there's going to be bacterial growth on this, there will be bacterial growth on this. Whether or not there's any bacteria that break down blood, I kind of doubt it, but fun to see. So some bacteria that do break down red blood cells in this media would be your Staphylococcus aureus, Streptococcus pyogenes that causes strep throat, different organisms that tend to be, usually can be pathogenic. And then for this last plate, I'll just do for the U factor, I'm just going to plate a bunch of the bacteria on the entire surface, not trying to isolate anything. And again, ESN methylene blue, looking for coliforms. Like I mentioned before, I see bees and manure piles and stuff like that. They collect water from pig wallows and stuff like that. So there's always a possibility there can be some gram negatives, but from my previous tests, I didn't see anything, so not really expecting anything, but we will find out. And then mannitol salt agar is a selective media and differential media for salt tolerant organisms and organisms that can actually ferment mannitol, a type of sugar. Since it's honey, again, not expecting any growth, but just for fun, we're going to plate it. Organisms like Staphylococcus aureus like to grow on this. It's a salt tolerant organism. and Staph aureus actually turns this media yellow if it's present due to the fermentation of mannitol. But anyway, we're plating it and having some fun, seeing if anything grows on that media. Alright, so I got a couple general purpose medias and a couple different medias that we're growing the honey on. Check back in 24 hours, see what they look like. Okay, so we're looking at the results from the honey experiment, and actually there is, hard to tell, but there is something growing on this eosin methylene blue plate. So that's this EMB again selects for gram-negative coliforms, and this isn't anything that I've ever seen as far as a common coliform that we work with, but I guess it's not surprising that there's a gram-negative growing there. It doesn't necessarily mean anything bad, but it's got a strange shape to it, the colony. Hard to see. Get in the right light so you see that kind of this area here. Something's growing there. But again, I cultured that that honey, so there would have been lots of that bacteria in that initial test tube that was turbid that I used as the inoculum. But mannitol salt auger, like I said, I wasn't really expecting anything on that just because we're culturing from honey, so there's nothing growing on this media. And then this is the streak plate from the honey. And again, this looks kind of gross, like there's a lot of bacteria, but when you have a turbid broth tube, there's millions of bacteria in there. So I do have isolation of this organism here, and it looks like a common bacillus organism, something that we work with in the lab. It's it's more of an environmental soil bacteria. I'll stain it. I'll show you what it looks like under the microscope. But bacillus organisms, they are capable of forming endospores, and endospores are a dormant structure that can basically survive long periods of time with nutrient deprivation. They tend to be heat stable, UV light stable. Some organisms are clinically significant, like Bacillus anthracis, for example, that causes anthrax. So I imagine this is probably Bacillus subtilis, which is a common soil organism, which is what I would expect that to be, and it looks like that just from my experience with working with that organism. But I'll do a gram stain of it, and I'll show you what it looks like with the microscope. This is the blood auger plate, and like I mentioned earlier, if I had growth on TSA, I would have growth on blood auger. But blood auger looks for hemolysis, so lysis of red blood cells. And this is the same organism growing on the media. I can tell that just by looking at it. And this is what we refer to as gamma hemolytic. There is no hemolysis, so that's another clue that this is a Bacillus subtilis and not anything that's like a pathogenic bacillus. And this is the U factor plate. So this is where I just spread the bacteria all around, and you can see that it's all that same bacillus organism. So raw honey, it does have bacteria in it, and again, from my initial test where I was trying to count bacterial numbers, the bacterial numbers in the raw honey are very low, less than 300 colony forming units per gram of honey. So this isn't an unexpected result. I would have expected there to be some bacteria in the honey, and this is just showing after culturing and growing the bacteria in a nutrient-rich media, these bacteria there. So these bacteria, like I said, the bacillus, they were hanging out in the endospore form. The concentration of the sugar in honey inhibits bacterial growth, so it's not actively growing in the honey. So that's why it doesn't go bad. Like I said, this honey was a year old. I still have honey from a year ago that we consume. And like I said, if this is a Bacillus subtilis common soil organism, it does not make you sick. But we'll do a little stain of this, show you what it looks like at 1,000 times magnification. And I'll actually, so the gram stain is what we call a differential stain. We differentiate between gram-positive and gram-negatives. So gram-positive organisms would be like a bacillus organism, Staphylococcus, Streptococcus, things that you may have heard of before. Gram-negatives are your E. coli's and salmonellas, among a bunch of other types of bacteria. The difference is their cell wall. So gram-positives have what we call a thick cell wall, and gram-negatives have a thin cell wall, just to keep it simple. So I'm just going to put a little bit of DI water on the slide here, just a little bit. Got a sterile loop, and I'm going to pick just a small amount of that isolated colony off the plate. I don't need a lot, just a small amount. And I'm going to put this on the slide. And I'm going to let those air dry there for a moment. I'm going to try taking some of this off of this eosin methylene blue plate. Let's see what we come up with here. And we gotta let those air dry. So I should mention, like I said, this kind of looks gross, but that isolated colony there that I'm staining, that was one bacterial cell that formed that colony. So there's maybe 3-4 colonies here in this area. They're not isolated from each other. In reality, there's not really a whole lot of bacteria in this area. It's just that bacteria, the colony morphology of it, when it has that space for and nutrients, so it's not competing with other organisms. Like over here, there's a lot of bacteria, a lot of cells, so they kind of bunch together and they're competing for nutrients. So this is one of the reasons why we can't quantify when you do a streak plate. But that one colony there was formed by one single cell. So it may look like it's gross and you wouldn't want to eat the honey, but again, I inoculated the honey in a broth tube. There were millions of bacteria in that broth tube that was turbid, and then I plated that on there and spread them out so that I would have isolated colonies. So it looks bad, but I can assure you that it is not, and the honey is completely fine to eat. So the risk for consuming raw honey is basically non-existent for those that have a strong immune system. You may have heard of infant botulism. That's caused by Clostridium botulinum, the same organism that can cause botulism from home canned goods. So that's commonly found in the soil. And with honey, the endospore, so Clostridium is an endospore forming bacteria again, that endospore can lay dormant in the raw honey, and then with children under 2, that bacteria has the ability, since they're considered immunocompromised when you're very young, to germinate and cause disease in very young. It's not common, but that is one of the risks associated with consuming raw honey. But for normal, healthy adults and stuff like that, our immune systems wouldn't allow for that organism to germinate and propagate and cause botulism. It's a toxin that's produced by the Clostridium botulinum that it causes the illness. So I kind of talked about the home canning risk with botulism in my raw milk study that I did. A little experiment that I did. That those botulism endospores can survive the heat process of home canning and can germinate inside the canning jar and grow and form that toxin in the jar. So if the food's not reheated, that like I mentioned, salsa, that toxin is not deactivated, and then that's what leads to the illness. So if it's something that you can and you reheat, the risk is low for that. But that is one of the organisms that can cause food spoilage in home canned goods, that if you do not properly can the food according to USDA guidelines, either pressure canning or water bath canning. Depends on the food. So I won't go into any more of those details. But all right, so I got some slides I'm staining here. The first step of the gram stain is to flood the slide with crystal violet. And crystal violet penetrates all cells, gram positive or gram negative. You let that sit on there for about a minute, and it doesn't matter if they're gram positive or gram negative, all cells at this point would stain purple. So it all has to deal with, this process has to deal with the thickness of the cell wall. So gram positives have really thick cell walls, gram negatives have thin cell walls, just to keep it simple. So you rinse that off. And then you add the grams iodine. Grams iodine is basically iodine that binds to crystal violet and makes the molecule larger. We call it the crystal violet iodine complex. So it turns the crystal violet molecule, maybe into a small size, like the size of a golf ball, into the size of a softball, just as an example. So you let that sit on there for about a minute and then rinse that off. And next is the most important step of this process, is a decolorizing step. So you take alcohol acetone mix and you take that, run it over the slide here, and the goal is to remove the crystal violet from gram negatives. So the decolorizing step, you can imagine a thick cell wall, gram positive, gram negative, thin cell wall. The crystal violet's going to rinse out of the gram negatives much quicker. Gram positives will retain that because it's basically like a really thick filter that crystal violet's trying to go through now. So at this point, if it's gram negative, those cells are left colorless. And the gram positives, they retain that crystal violet. Since the gram negatives, if you have a gram negative, would be colorless, we counter stain with safranin, and that would stain gram negative cells. So gram positive cells end up being purple, gram negatives end up being pink. You let that set up for about a minute, and you rinse that off. Now, like I mentioned, I imagine that one organism from the TSA plate is probably a gram-positive bacillus, and we will confirm that with the microscope. Alright, so I got my digital microscope set up here. We'll look at this first slide. First we got to get it into focus at a lower power, so 100 times magnification. So this is 100 times magnification, and I'm already seeing bacillus shape. So bacillus means rod shaped. So we'll change this to 40x, which is 400 times magnification. There we go. Alright, so now we see those rod-shaped bacteria. 400 times magnification. And then we'll do what's called oil immersion. So I put some oil right on the slide. And then see them at 1,000 times magnification. Alright, so this is what they look like at 1,000 times magnification. So we see these kind of rod, almost rectangular shaped cells, and they are usually linked together like this. If you have them in a broth culture, then they probably would seem more connected. But classic looking bacillus organism. Like I said, probably Bacillus subtilis, which is a very common soil microbe. The colony morphology, what it looked like on the plate, looked just like it, and the cell morphology looks just like that as well. So a completely harmless bacteria that's found in the honey. And again, that remains dormant in the honey. It's not growing in the honey. It just can then germinate when it has optimal conditions. Since I was curious as to why that bacteria was growing on EMB, I did take some and put it on TSA plate, and it turns out it had the same colony morphology as the other organism. So it is a gram-positive rod, the same organism, and it was just able to grow on the ESN methylene blue. So we'll look at that other slide from the ESN methylene blue.
Appalachian sunrise meets my skin.
This appears to be a gram-positive that was growing on that as well. It might be the same organism. So, like I said, ESN methylene blue usually inhibits most gram-positives, and we say most gram-positives. If there's a lot on there, then maybe they will grow. This appears to be something different, though, than the previous slide. The cell morphology here, it's thinner, not as boxy rectangular. So I think it is a different species growing on that ESN methylene blue. And they seem to be more connected here. So this would be multiple cells connected to each other. So that's interesting that they were growing on that media. But like I said, it's a selective and differential media for gram-negative coliforms. This is not a gram-negative coliform. And this just shows you that science isn't always 100% correct as far as when we have these media. There's always an exception for the rules that we apply. But in general, again, this is probably something that's harmless, could be from the Bacillus genus, endospore forming, and is not propagating, not growing in the media. But interesting nonetheless. So I kind of outlined some of the risks involved with raw honey with young people. This little experiment shows that there is some bacteria in raw honey. Very low numbers from the honey that I have. Because I wasn't able to obtain a countable plate, I had to actually put that sample into a nutrient media and grow those few cells into lots of cells in order for them to then plate and then pick a colony to actually stain. So Anyway, hopefully you enjoyed watching this little experiment with raw honey. Again, I'm not trying to scare people off from consuming it. That U factor plate was just, lots and lots of bacteria growing on that. It takes one single cell to form one of those colonies. And just depending on the bacteria, some bacteria morphologies can be very large, like what we saw there, and some can be very tiny, like what was on that ESN methylene blue plate. So, anyway, hopefully you enjoyed. Hit the thumbs up. We'll see you next time.